the lecture on fatty acid synthesis, and this is the last lecture concerning fatty acids or metabolism of fats. Now, in the previous lectures we have looked at beta oxidation of fatty acid, a process that is primarily aimed at breaking down fatty acids into acetyl coenzyme A. In this particular lecture, what we are going to do is to look at the synthesis of fatty acid, and the process also requires the presence of acetyl coenzyme A. So if we want to have a roadmap for this lecture, what we are going to do here is first to look at how carbohydrate metabolism, which you have studied in quite a bit of detail, is related to fatty acid synthesis. And then we will talk about the overview of fatty acid synthesis, and this particular part is very important for your assessment. Then we will talk about how acetyl coenzyme A is important for the process of fatty acid synthesis. We will then look at the rate limiting step of fatty acid synthesis, also very important for your assessment. We will have a discussion on the fatty acid synthase enzyme, and this particular aspect is also important for your assessment. In this lecture, in the recording, of course, I have gone into the details of the fatty acid synthetic process, but for your assessment only a general overview is required, so you do not need to remember the chemistry involved in the process of fatty acid synthesis, although I have discussed it so that you have a fair bit of understanding of the process. During the process of synthesis, we will talk about different cofactors and prosthetic groups. Again, this part is extremely important for your assessment because deficiency of the cofactor or a prosthetic group impairs the process of fatty acid synthesis, and this particular aspect is therefore clinically relevant. The major aim of the process of fatty acid synthesis is to synthesize palmitic acid or palmitate, and in this lecture I will be highlighting what is the importance of palmitate. After highlighting the importance of palmitate, we will have a brief discussion on COX inhibitors. Again, this is extremely important. I believe this particular concept will be revisited in your course of pharmacology in phase 2, and we will conclude the lecture by looking at how amino acids or amino acid metabolism contributes to the process of fatty acid synthesis. So this is the roadmap that we will follow. I highlighted the sections which are important for you for your assessment, so please take a note of those particular points, and I will urge you to use a notebook while you are going through this particular lecture, because in the class I will only present a summary of this lecture, and then we will have a discussion around the important aspects related to fatty acid synthesis. So let us start. As I told you, the process of carbohydrate metabolism is intricately related or associated with fatty acid synthesis, and this particular slide actually integrates the process of carbohydrate metabolism with the process of synthesis of fats. Now, one of the important requirements of fatty acid synthesis is NADPH, and in your course you have looked at the pentose phosphate pathway, where glucose is converted to ribulose 5-phosphate, and in the process of this conversion you generate NADPH. Of course, NADPH is important for the homeostasis inside the erythrocytes, and you have looked into this process in quite a bit of detail. But this NADPH is also a key requirement for the process of fatty acid synthesis. Now, glucose is converted to pyruvate, and pyruvate is then converted to acetyl coenzyme A. And this process of conversion of pyruvate to acetyl coenzyme A is catalyzed by the multi-subunit enzyme, the pyruvate dehydrogenase complex. You have looked at that when we talked about TCA cycle. And this acetyl coenzyme A is the precursor for the formation of fatty acid. So without the TCA cycle, you will not have fatty acid synthesis inside the physiological environment. When we were talking about the gluconeogenetic process, the gluconeogenesis pathway, we focused on the malate oxaloacetate shuttle, and the malate oxaloacetate shuttle is actually responsible for the conversion of malate to pyruvate. This you already know. But what you probably wouldn't have focused on is that when malate is converted to pyruvate, you also generate NADPH. So I would like you to take a note of this, that apart from the pentose phosphate pathway, the malate oxaloacetate shuttle during gluconeogenesis is also able to generate NADPH, and this NADPH is again an important or essential factor for the synthesis of fatty acid. So in summary, the carbohydrate metabolism inside our body provides two essential factors which are required for the synthesis of fatty acid. One is NADPH, which is produced by the pentose phosphate pathway and the malate oxaloacetate shuttle. And the second one is acetyl coenzyme A, which is produced from glucose through pyruvate by the participation of the glycolytic pathway and then the pyruvate dehydrogenase complex. Now, citrate is also converted to acetyl coenzyme A, and this particular process we will look in this lecture in a few slides. So, in summary, what I can tell here is fatty acid synthesis requires the cooperation of several metabolic pathways, especially the pathways that are related or associated with carbohydrate metabolism. Towards the end of the lecture, we will also see how amino acids feed into the fatty acid synthesis process, but predominantly carbohydrate metabolism generates the essential precursors and factors that are required for the synthesis of fatty acid. Now, this particular slide talks about the overview of the process of fatty acid biosynthesis. Very, very important for your assessment. So what you need to know is the process of fatty acid synthesis occurs in the cytosol. So remember that although you are using acetyl coenzyme A, and most of the processes that you have looked into so far where acetyl coenzyme A is involved occurs in the mitochondrial matrix. But fatty acid synthesis is a metabolic pathway which, although involves acetyl coenzyme A, it still occurs in the cytosol. Now, in human beings or any animal or any organism, belonging to the animal kingdom, you will have this particular process occurring in two predominant organs. One is the liver or the hepatocyte, and second is the mammary gland. However, if someone asks you what is the predominant site for the synthesis of fatty acid, the answer is the liver. The process of fatty acid synthesis uses two important metabolites. One is the acetyl coenzyme A, and the acetyl coenzyme A is predominantly derived from the metabolism of carbohydrate, which I showed you in the previous slide. And the second metabolite that is used in the process of fatty acid synthesis is NADPH, which is predominantly generated from the pentose phosphate pathway and to some extent from the malate oxaloacetate shuttle. The aim of fatty acid synthesis is to produce palmitic acid, which is further modified to produce other types of fatty acid. And we will look at some of these reactions which help the processing of palmitic acid into other types of fatty acid or important metabolites. So this is like a general overview which talks about the site of fatty acid biosynthesis and what are the important metabolites that you require for the process of fatty acid biosynthesis to occur in the physiological milieu. The aim of fatty acid synthesis is to synthesize a 16 carbon long fatty acid, and that is palmitic acid. And after the formation of palmitic acid, it is further modified or processed to form other important fatty acids or important metabolites, which we will discuss in the subsequent slides. So, as I mentioned, the process of fatty acid synthesis happens in the cytosol, predominantly in the hepatocytes or the liver cells. Now, as I told you, most of the metabolic processes that you have encountered so far happens in the mitochondrial matrix, and acetyl coenzyme A therefore has to be transported from the mitochondrial matrix into the cytosol, and the process is not very straightforward. So first what happens is you have acetyl coenzyme A, and you can see here in the figure that it is indicated that acetyl coenzyme A can be generated from two important biomolecules. One is glucose. So glucose is converted to pyruvate by the process of glycolysis, and pyruvate by the catalytic activity of the enzyme pyruvate dehydrogenase complex is converted to acetyl coenzyme A. And this conversion of pyruvate to acetyl coenzyme A happens inside the mitochondria. Similarly, as we shall see towards the end of the lecture, amino acids, well, more precisely specific amino acids, also give rise to acetyl coenzyme A. And this acetyl coenzyme A, of course, is present inside the mitochondrial matrix. So the first step in the process of fatty acid synthesis is to convert this acetyl coenzyme A to citrate. So acetyl coenzyme A is converted to citrate by the enzyme citrate synthase. And once citrate is formed, as you can see here, through the tricarboxylate transporter, citrate migrates from the mitochondrial matrix to the cytosol. So in the assessment, if somebody asks you which of the following metabolites will not be formed if the tricarboxylate transporter is inhibited, you should look for palmitic acid or a fatty acid, because without citrate migrating from the matrix to the cytosol, the process of fatty acid synthesis cannot start. So coming back, citrate through the tricarboxylate transporter passes into the cytosol. Now in the cytosol, citrate has to be converted to acetyl coenzyme A, because acetyl coenzyme A is the precursor molecule for the synthesis of fatty acid. And the enzyme that catalyzes the conversion of citrate to acetyl coenzyme A is citrate lyase. And you can see here the process of conversion of citrate to acetyl coenzyme A is ATP dependent. That means synthesis of fatty acids require the presence of ATP, which tells you that only when the physiological system has an excess of ATP, only then will the body synthesize fatty acid. Because if I have decrease in the number of ATP molecules, I will try to replenish the ATP stores, and then glucose will be channeled more towards the catabolic pathways, where it will be converted to pyruvate, and pyruvate under aerobic or anaerobic conditions will generate ATP. So citrate will be converted to acetyl coenzyme A in the presence of ATP by the enzyme citrate lyase. And once it is converted to acetyl coenzyme A, this particular process of fatty acid synthesis will be initiated. Well, when citrate is getting converted to acetyl coenzyme A, it also produces oxaloacetate, and oxaloacetate is then converted to malate, as you can see here by the enzyme malate dehydrogenase. Now malate has two fates. One, malate is converted to pyruvate by the enzyme malic, or malate is directly transported back inside the mitochondrial matrix by the malate alpha ketoglutarate transporter, where in the presence of oxaloacetate, it is converted back to citrate, and this particular reaction you already know. The pyruvate that is produced in the cytosol is taken up by the mitochondria, and you already know this. This is the translocase, the pyruvate transporter, and it also migrates inside the mitochondrial matrix, where it may be converted to oxaloacetate by the presence of the enzyme pyruvate carboxylase, and this particular process also requires ATP. So the whole process of transport of citrate from the mitochondrial matrix to the cytosol and the conversion of citrate to acetyl coenzyme A is an ATP dependent process, as well as once oxaloacetate is converted to malate to pyruvate and the pyruvate is transported to the mitochondrial matrix, the process of conversion of pyruvate to oxaloacetate also requires the presence of ATP. So the whole process, the whole shuttle that we are looking here, or that we are trying to get an idea about, requires quite a bit of energy. So the process of fatty acid synthesis will occur only when body has enough reserves of ATP. And I mentioned this earlier when I was discussing this particular step in the process of fatty acid synthesis. So at the end of this particular step, you have been able to transport citrate from the mitochondrial matrix inside the cytosol, and you have been able to convert citrate to acetyl coenzyme A. And this acetyl coenzyme A will now act as the precursor for the synthesis of fatty acid. Now, what I want you to remember is that the main or the key step, which is also the rate limiting step, which we shall see in a minute, in the process of fatty acid biosynthesis is the conversion of acetyl coenzyme A to Malonyl coenzyme A. And if I can say here, Malonyl coenzyme A is an activated form of acetyl coenzyme A, which is used for the synthesis of fatty acid. So the key step in the formation of fatty acid synthesis is the formation of Malonyl coenzyme A from acetyl coenzyme A. So let us now try to see the process of conversion of acetyl coenzyme A to Malonyl coenzyme A in a bit of detail. Before we go there, let me tell you a fact, and this you have to remember, that when a cell or organism has more than enough metabolic fuel available to meet its energetic needs, the excess is generally converted to fatty acid and stored as lipids such as triacyl glycerol. And by now you know what triacyl glycerol is. Now if you look at the process, and we looked at this shuttle in the previous slide, in the first step you have taken the citrate out of the mitochondria to the cytosol. Once citrate comes inside the cytosol, the enzyme citrate-lyase converts it to acetyl coenzyme A. And acetyl coenzyme A is then converted to Malonyl coenzyme A. Before I go to this step, let us look at the control of the conversion of citrate to acetyl coenzyme A. This particular step is under the tight control of the hormone insulin. What insulin does is that it triggers the phosphorylation of citrate-lyase, and phosphorylation of citrate-lyase activates the enzyme. As a result, it converts citrate to acetyl coenzyme A. You can see here the pyruvate dehydrogenase complex is also activated by phosphorylation by the hormone insulin, and this particular step we discussed in detail when we took the citric acid cycle. As pyruvate dehydrogenase complex is activated, increased quantities of pyruvate are converted to acetyl coenzyme A. So insulin favors the formation of acetyl coenzyme A either from citrate or from pyruvate. Now once we have acetyl coenzyme A and we have enough ATP, the acetyl coenzyme A will be first converted to Malonyl coenzyme A, which is the activated form of acetyl coenzyme A. And this particular step is catalyzed by the enzyme acetyl coenzyme A carboxylase. Now, malonyl coenzyme A through successive steps will be converted to palmitoyl coenzyme A, and we shall look at these steps in a moment. But let me come back to the step of conversion of acetyl coenzyme A to malonyl coenzyme A. As I mentioned, the conversion of acetyl coenzyme A to malonyl coenzyme A is catalyzed by the enzyme acetyl coenzyme A carboxylase. Now, the acetyl coenzyme A carboxylase is under the tight regulation of two hormones, glucagon and epinephrine. Both these hormones trigger the phosphorylation of acetyl coenzyme A, as a result of which the enzyme gets deactivated and acetyl coenzyme A cannot be carboxylated to malonyl coenzyme A. In summary, therefore, glucogenic hormones, that means hormones that increase the level of blood sugar, such as glucagon and epinephrine, prevent the synthesis of fatty acid. Also, once we have significant amounts of palmityl coenzyme A, palmityl coenzyme A allosterically inhibits the enzyme acetyl coenzyme A carboxylase. So, it is a form of product inhibition. Although the product here is not malonyl coenzyme A, but the ultimate metabolite that is generated from the process of fatty acid synthesis. So, coming back, the reaction of the process of conversion of acetyl coenzyme A to malonyl coenzyme A is catalyzed by the enzyme acetyl coenzyme A carboxylase, and for you this is very important, which is the rate limiting step in the biosynthesis of fatty acid. And this enzyme is an important site of regulation in vertebrates, that means in human beings. Palmitoyl coenzyme A, the principal product of fatty acid synthesis, acts as a feedback inhibitor of the enzyme, and citrate is an allosteric activator of the enzyme. So, this particular process is activated by citrate, as you can see here, but inhibited by palmitoyl coenzyme A. Also, glucogenic hormones such as glucagon and epinephrine also inhibit the process of carboxylation of acetyl coenzyme A. So, this is the rate limiting step. So, you need to remember here that the rate limiting step of fatty acid synthesis is catalyzed by the enzyme acetyl coenzyme A carboxylase, and this particular enzyme has two allosteric regulators. Citrate is an allosteric activator, whereas palmitoyl coenzyme A is an allosteric inhibitor. Now, the other points to remember here is that the process of conversion of acetyl coenzyme A to malonyl coenzyme A requires biotin, or more specifically vitamin B7. Before we go into that, let us try to look at some of the important features of this particular step. Formation of malonyl coenzyme A from acetyl coenzyme A is an irreversible process. That's the reason if you see here, the process is shown with an unidirectional arrow. The conversion, which you now know is catalyzed by a carboxylase, more specifically acetyl coenzyme A carboxylase, which contains biotin as a prosthetic group. You should know what a prosthetic group is because this has been discussed in detail when we talked about enzyme structure function. So, deficiency of biotin or vitamin B7 leads to impaired function of acetyl coenzyme A. Remember, we talked about another carboxylase, pyruvate carboxylase, and I mentioned that egg whites contain a protein that bind to biotin and inhibit its activity. So similarly, increase in avidin in the physiological environment will also inhibit or impair the activity of acetyl coenzyme A carboxylase. So this is clinically relevant information. I will urge you to take a note of this. So there are two enzymes that require biotin as the prosthetic group in your course. Both of them are carboxylases, and therefore avidin, a protein that tightly binds to biotin, will impair the activity of these carboxylases. Coming to this figure, if you look at it, this is biotin, which is linked to a carrier protein called biotin carrier protein, and this carrier protein has a side chain, and this side chain through which it binds to biotin involves the amino acid lysine. So if you have deficiency of lysine, synthesis of the biotin carrier protein will be impaired, as a result of which biotin will not be available to these two carboxylases. In our case now it is acetyl coenzyme A carboxylase, since we are studying fatty acid synthesis. As a result, acetyl coenzyme A cannot be converted to malonyl coenzyme A, and consequently therefore fatty acid synthesis will be impaired. So take a note of this particular amino acid lysine that is important for the presentation of biotin to the carboxylase. You can of course ignore this part of the figure. I believe next week when Prof. Riyadh starts talking about integration and regulation of metabolism, he will talk in detail about the different hormones and the process by which these hormones regulate the process of metabolism. But for your information, you need to know that acetyl coenzyme A carboxylase is regulated tightly by hormones. I already mentioned about glucagon and epinephrine, that glucagon and epinephrine promote the carboxylation of acetyl coenzyme A carboxylase, as a result of which the enzyme becomes inactive. But I didn't mention in that particular slide about the dephosphorylation. Now, dephosphorylation of acetyl coenzyme A makes it active. So the hormone which promotes fatty acid synthesis, because this is a hormone which reduces blood sugar, so it will promote anything that will help in the clearance of blood sugar. In other words, insulin will promote fatty acid synthesis. In order to promote fatty acid synthesis, it has to dephosphorylate acetyl coenzyme A carboxylase, and the process of dephosphorylation leads to the activation of the enzyme, and once the enzyme is activated, the process of fatty acid synthesis is initiated. Dephosphorylation, activation mediated by insulin, phosphorylation, inactivation mediated by two key hormones, glucagon and epinephrine, which are glucogenic hormones secreted in our body. Often in the different licensing exams, they ask about the secondary messengers that are involved. So glucagon and epinephrine are cyclic AMP dependent protein kinases. That means they activate cyclic AMP dependent protein kinase, which promotes the phosphorylation of acetyl coenzyme A carboxylase. Insulin, on the other hand, activates protein phosphatase, which dephosphorylates acetyl coenzyme A carboxylase, activating it. Probably Dr. Bakroom would have talked about the secondary messengers when he talked about cell signaling. So I am not going to go into the details of this in this particular lecture. Now we come to the second most important enzyme. Of course, please remember that this particular enzyme, that is the fatty acid synthase enzyme, is not catalyzing the rate limiting step in fatty acid synthesis. Very often people make this mistake. So please take a note of this. The rate limiting step in fatty acid synthesis is not catalyzed by the fatty acid synthase enzyme, but it is catalyzed by acetyl coenzyme A carboxylase. Now this is the fatty acid synthase enzyme, and we will see how this enzyme acts in the synthesis of fatty acid. We will discuss these steps in a moment, but what I will like you to highlight to you is that although this is a single polypeptide, the single polypeptide has different enzymatic functions. This is not the quaternary structure, this is the tertiary structure of the protein. The different colored segments identify the different enzymatically active regions. And what I will also like to highlight here, if you look at it, the process of fatty acid synthesis essentially requires NADPH, and this NADPH is coming from pentose phosphate pathway. Very, very important. In general, I will say that this is the most important slide for you, because the process of fatty acid synthesis involves condensation of an activated acyl group, and an activated acyl group is an acetyl group from the acetyl coenzyme A molecule, and condensation of an activated acyl group involves two carbons from the malonyl coenzyme A molecule that you have generated by the catalytic activity of the enzyme acetyl coenzyme A carboxylase. In the process, there is an elimination of one carbon dioxide molecule, and that's a decarboxylase. So you are losing one carbon dioxide, and this extends the malonyl coenzyme A by two carbons. Essentially, the process is involving reduction, so reduction by NADPH, and dehydration, that means loss of water. And as I mentioned, NADPH comes from the pentose phosphate pathway. This is all that you need to know for fatty acid synthesis in terms of its mechanistic overview. But we need to go through the individual steps, and as I told you, I'm not going to go through the steps during the lecture. You have to listen to the recording, because the individual steps involve specific cofactors and prosthetic groups, and these cofactors and prosthetic groups therefore are clinically relevant. So as I told you, fatty acid synthase has 7 different enzyme activities. Now many people make a mistake that they also teach that, and this is why I mentioned this here. The fatty acid synthase in case of human beings consists of one large polypeptide with 7 different activities. For example, you have ER, meaning enoyl reductase. But in case of lower organisms such as bacteria and plants, the fatty acid synthase consists of seven different polypeptide chains. In case of human beings, one polypeptide chain. In case of bacteria and lower organisms, 7 polypeptide chains. So here we will observe quaternary structure. Here we will observe only tertiary structure. Don't make this mistake. And also, I have seen many people teaching it, telling that the fatty acid synthase consists of 7 different polypeptide chains in human beings. So in human beings, it is not the case. In human beings, we have one polypeptide chain with 7 different activities. Of course, in between bacteria and human beings, we have an organism called the yeast. And as you can see here, in case of yeast, there are 7 activities but 2 different polypeptide chains. So as evolution has happened, the number of polypeptide chains has decreased, and however, the activities have been conserved. The most important component of the fatty acid synthase enzyme is the acyl carrier protein, also known as ACP. You can see this in this particular schematic, and now we have to study why is this particular protein important. Very important for a short answer or short note type of question. Now the function of acyl carrier protein is that it helps in the tethering of the growing fatty acyl chain to the fatty acid synthase enzyme. Now this is the ACP, and as you can see here, the acyl carrier protein requires a prosthetic group, and that particular prosthetic group is pantothenic acid. Therefore, I can say that pantothenic acid promotes the synthesis of fatty acid, or more precisely, palmitic acid or palmitate. Now, we already know that insulin binds to a specific receptor called the tyrosine kinase receptor. We talked about this when we talked about glucose transporters during our initial lectures on glycolysis. So coming back, pantothenic acid promotes the synthesis of palmitic acid. Palmitic acid synthesis leads to acylation of tyrosine kinase receptor. Or if you do not remember the word tyrosine kinase, at least remember that palmitic acid favors the acylation of the insulin receptor. Acylation of tyrosine kinase receptor or insulin receptor leads to decreased binding of insulin to tyrosine kinase receptor. That means if I acylate the receptor of insulin, there will be decrease in the binding of insulin to its receptor. Now let us take a hypothetical condition where pantothenic acid is absent or the levels of pantothenic acid is decreased. If I decrease the level of pantothenic acid, I will have a decrease in the synthesis of palmitic acid. Therefore, I will have a decrease in the acylation of insulin receptor. Since there is less of acylation, therefore there will be increase in insulin binding to its receptor, increase in the binding affinity of insulin receptor to insulin will increase the number of glucose transporters. If I increase the number of glucose transporter on the cell, there will be decrease in the level of blood sugar. Therefore, this will lead to hypoglycemia. In other words, pantothenic acid deficiency leads to hypoglycemia. Very, very important. What I will ask you to do is to write the answer for a short answer question, and the question is as follows: why does deficiency of pantothenic acid lead to hypoglycemia? So we move on and we try to look at the chemistry of the process of fatty acid synthesis. As I told you, these steps are not really important when it comes to the process of assessment. And when I mean important, meaning in relation to the mechanism that is involved, but what is important for you is to look at the cofactors or the prosthetic groups that are involved in the process. So in step 1, acetyl coenzyme A. So you have acetyl coenzyme A in the cytosol and the process is now beginning. In this first step, acetyl coenzyme A is loaded onto the fatty acid synthase. And you can see here, this is acetyl coenzyme A. And these are the 7 different activities which I referred to when I talked about the fatty acid synthase enzyme. I will request you not to remember these different activities, not important, and please don't try to memorize them as well. So what I want you to look here is, this is the fatty acid synthase enzyme, and you can see here in the first step, acetyl coenzyme A is loaded onto the fatty acid synthase enzyme. In the next step, I need acetyl coenzyme A and I need activated form of acetyl coenzyme A. In the first step, I loaded acetyl coenzyme A. In the second step, I have to load the activated form of acetyl coenzyme A, which is malonyl coenzyme A. Malonyl coenzyme A has been generated earlier by the catalytic activity of the enzyme acetyl coenzyme A carboxylase. So in the second step, as you can see here, I load fatty acid synthase with malonyl coenzyme A. In the first step, I had acetyl coenzyme A, and this is the acetyl coenzyme A here, and then the second step we have loaded fatty acid synthase with malonyl coenzyme A. So there are 2 stages in the initiation phase. The first is to load with acetyl coenzyme A. Second is to load with malonyl coenzyme A. And as you can see here, the malonyl coenzyme A is loaded onto the acyl carrier protein, and therefore for this loading to happen, you require the presence of pantothenic acid, which I discussed in quite a bit of detail previously. Then we will have 4 consecutive steps: condensation, reduction, dehydration, reduction. Let us go through them very quickly. So first is a condensation reaction that is happening here. As you can see, in this process there will be a decarboxylation happening. Carbon dioxide comes out, and here you get the alpha and the beta carbon for your fatty acid, or in this case palmitic acid. And then you have a reduction, and you can see here, because of the reduction you have a hydroxyl group that is formed, and then we progress on to the next step where there is dehydration happening, loss of water, and then there is another reduction happening. And as you can see here, you get a saturated acyl group now, but lengthened by 2 carbons. So if I go back, there was 1 carbon here. At the end of it, you have 2 more carbons that have been added. Process involves decarboxylation. So this is decarboxylation, reduction, dehydration, and reduction again. So these are the 4 steps that will happen, which will help in the formation of a saturated acyl group, which is lengthened by 2 carbons. As I told you, please do not focus on the mechanism. Why I focused on these particular slides is that to give you a parallel of the process in terms of beta oxidation. So you can see there was also the similar steps that were occurring, opening and closing of double bonds, hydroxylation and oxidation that was occurring. In this case, reduction is occurring. So beta oxidation involves oxidation, whereas fatty acid synthesis involves reduction. So we go through the steps again quickly. Reaction of malonyl group with acetyl group to form acetyl CoA, acetoacetyl acyl carrier protein. If you look at this process here, acetoacetyl acyl carrier protein. So this is the acyl carrier protein. This is the acetoacetyl part. Condensation has happened with decarboxylation. So loss of carbon dioxide. One of the important byproducts of fatty acid synthesis, therefore, is carbon dioxide. Please make a note of this point. Next, after decarboxylation, we have reduction. The process of reduction requires NADPH. That NADPH is supplied by 2 processes, predominantly PPP, but to some extent by the malate oxaloacetate shuttle, as I previously mentioned. And you can see here, once I have reduced this particular acetoacetyl acyl carrier protein part, you have a hydroxyl group that is formed. Now there is dehydration. As there is dehydration, this particular hydroxyl group will be converted into a double bond and there will be a loss of water. Lastly, after dehydration, there is reduction again, and you can see here in the process of reduction, you have lengthened the acyl group by 2 carbons. The reduction process again requires NADPH, which predominantly comes from the pentose phosphate pathway. If I go back quickly, if you look at the process, you will see that at each step different parts of the enzyme are functioning. Again, going back, look here, first it was Kr part, then the Er part, then the DH part, then the Ks part, and this is how a single polypeptide chain, by having different domains, is able to divide the process of catalysis. So this is very, very important when it comes to the fatty acid synthase enzyme. In case of humans, it is consisting of a single polypeptide chain but has 7 different active sites and 7 different activities. Now, so we have ended this once we have formed the double bond, and once we have at the end, what we do is we close this and we transfer it. So this is what is happening. We translocate this to a particular part of the enzyme so that this acyl carrier protein is free to trap more malonyl coenzyme A, and the next cycle begins. So now at the end of the first cycle, you have a butyryl group. You know what is a butyryl group. Now then again the ACP takes up one malonyl coenzyme A, and the process of condensation, reduction, dehydration, reduction is occurring again, and you will at the end of it increase the length of this particular butyryl group by another 2 carbons. So if this process is continuously continued, you can see here at each step you are adding 2 carbons, and at the end of subsequent steps you will be able to generate palmitic acid, which is a 16 carbon containing fatty acid. The process of course requires the presence of water and NADPH, and in the process I am generating carbon dioxide. So fatty acid synthesis will generate the metabolite carbon dioxide, and the process of fatty acid synthesis will continue till palmitate is formed. Take home message: acetyl coenzyme A is the precursor. It is first activated to malonyl coenzyme A, and then there is condensation, and several steps of condensation catalyzed by the enzyme fatty acid synthase leads to the formation of palmitic acid. The rate limiting step, of course, in the process is not catalyzed by fatty acid synthase, but is catalyzed by the enzyme acetyl coenzyme A carboxylase. Now we have to look at what happens to palmitic acid. If I want to make fatty acids containing more than 16 carbons, what I will do is to elongate palmitate. So if I want to make higher order fatty acids, I will elongate the palmitate, and if I want to make unsaturated fatty acids, we talked about unsaturated, monounsaturated and polyunsaturated fatty acids. If I want to make desaturated or unsaturated fatty acids, I will desaturate the palmitate, and this process predominantly occurs in the endoplasmic reticulum. More specifically, it occurs in the smooth endoplasmic reticulum, and I think Dr. Nerissa would have talked in detail about the function of the smooth endoplasmic reticulum. Now the process of elongation, very quickly, is very similar to the synthesis process, where 2 carbon units are added from activated form of acetyl coenzyme A, that is malonyl coenzyme A. What about desaturation? The desaturation requires the presence of NADPH and a system of enzymes known as cytochrome B5. And in the process of desaturation, if you can see here, this is a cycle similar to what we studied in the glutathione cycle. NADPH is converted to NADP. In the process, it reduces the cytochrome B5 system of enzymes. The reduced cytochrome B5 system then is responsible for desaturating a saturated fatty acid, and you can see here in the process oxygen is reduced to water and you have one double bond. So where is the NADPH coming from? Again, the answer to this is the pentose phosphate pathway. So if I ask you a question on G6PD deficiency, you have to remember that apart from glutathione, the formation of unsaturated fatty acids will also be impaired in these people, in these subjects. Now we come to essential fatty acids. Essential fatty acids are those fatty acids which have to be supplemented in our diet because our body does not produce them in adequate amounts. There are two fatty acids, linoleic acid and linolenic acid. Linoleic acid is an omega-6 fatty acid. Remember, when we talked about the omega classification, we started counting from the methyl end. So 1, 2, 3, 4, 5 and 6. So this is an omega-6 fatty acid because you have encountered the first unsaturation at the 6th position from the methyl end or the reducing end. And this is an omega-3 fatty acid because this is 1, 2 and 3, and this is the omega end. So there are two fatty acids. One is the omega-6 fatty acid, linoleic acid, and the other one is a linolenic acid, which is not produced in our body in adequate quantities. They have to be supplemented through different forms of diet, and you can see here from the picture what forms of diet contain which of the following fatty acids. And once they are supplemented, the body is able to use these fatty acids for the synthesis of other metabolites. We have to focus, and I'm not going to really go into the details of this because first, we don't have enough time, and I do not have the time to discuss the chemistry of how linoleic acid and linoleic acid and linolenic acid and linoleic acid is important, but we will focus on one of the aspects, which is the conversion of linoleic acid to arachidonic acid. And why I'm going to focus on this, because the conversion involves a system of enzymes known as cyclooxygenase or COX. So here, if you see here, linolate or linoleic acid is a precursor for the formation of arachidonate, and this arachidonate, by the catalytic activity of the enzyme COX, is converted to either prostaglandins or thromboxanes. This is more clear in this particular part of the slide. Sorry if this slide is a bit of a loud one, but for your purpose, you just need to focus on these particular steps: linolate converted to arachidonate, which are converted to prostaglandins and leukotrienes. And this process involves a system of enzymes known as COX. So arachidonic acid is used to make prostaglandins, thromboxanes and leukotrienes. Now there are two types of COX, COX-1 and COX-2. COX-1 is, sometimes people call it the good enzyme, and COX-2 the bad enzyme, or more specifically good COX and good bad COX. COX-1 is responsible for protecting the stomach lining, decreases fever and promotes the formation of blood clot by facilitating the aggregation of platelets. I believe you now know what platelets are. Dr. Laila would have introduced you to them in the foundations course. Now, when a person is suffering from some kind of pain, the person most of the time takes aspirin, commonly available over the counter, and non-steroidal anti-inflammatory drugs such as ibuprofen, also available over the counter. The brand name for aspirin is already, you know, aspirin. For ibuprofen, it is sold under the brand name of Brufen. The problem with using these drugs repeatedly is that these are non-specific inhibitors of COX. That means they block both COX-1 and COX-2. That means they not only reduce pain and suppress inflammation, which is mediated by COX-2, but they also inhibit COX-1. And since they inhibit COX-1, the patient has the risk of loss of stomach lining or it may also delay clotting. Now, loss of stomach lining leads to ulcer, and that is the reason sometimes in the medical exams it is asked why the repeated use of aspirin increases the risk of ulcer. The answer to that is because it inhibits the enzyme COX-1, which is responsible for protecting the stomach lining. The other activity, prevention of blood clot, is used today or it has been harbored today for treating cardiovascular disorders, especially people who suffer from non-ventricular atrial fibrillation. Before they prescribe NOACs and other forms of anticoagulants, the clinician will recommend a form of aspirin which is sold in the market under the brand name aspirin cardio or sustained release aspirin, and the sustained release aspirin prevents blood clotting by delaying or inhibiting the process of platelet aggregation. However, repeated use of aspirin or such form of aspirin increases the risk of gastric ulcer or loss of stomach lining. In order to treat pain, therefore, we have the COX-2 inhibitors which do not affect COX-1, and the COX-2 inhibitors, the specific ones, they protect the stomach lining and promote blood clotting, but since they block COX-2, they reduce pain and suppress inflammation. And any kind of coxib that is available in the market is a COX-2 inhibitor. The most prescribed coxib is celecoxib under the brand name Celebrex, but there are other forms of coxib drugs such as rofecoxib. I do not know if this is available here in UAE, but celecoxib is. I also don't know if valdecoxib is available in the UAE, but celecoxib is definitely available, and celecoxib is a specific COX-2 inhibitor, so the associated risk that is involved in taking non-steroidal anti-inflammatory drugs is reduced when you use specific COX-2 inhibitors. So the take-home message here is, going back to the previous slide, COX is responsible for the conversion of arachidonic acid to prostaglandins or thromboxanes. There are two forms of COX, COX-1 and COX-2. COX-1 is a good COX, COX-2 is a bad COX. COX-1 is involved in the protection of the stomach lining, and I mentioned why therefore regular ingestion of non-specific COX inhibitors increases the risk for gastric ulcer. Of course, we talked about aspirin in quite a bit of detail when we talked about enzyme inhibition. Please try to remember that and try to now bring these concepts into focus, and that is the reason I mentioned why certain parts of this lecture are of increased or augmented clinical relevance. We try to end the lecture now with how amino acid contributes to fatty acid synthesis. Very specifically, if I want to look at it, amino acid degradation leads to acetyl coenzyme A or citrate, and you can see that in this particular part of the slide. Isoleucine, leucine, threonine and tryptophan lead to the generation of acetyl coenzyme A. Alanine, cysteine, glycine, serine, threonine and tryptophan lead to the formation of pyruvate, which is ultimately converted to acetyl coenzyme A. And certain other amino acids feed into different parts of the TCA cycle, and they are ultimately also converted to acetyl coenzyme A. And acetyl coenzyme A, once it is converted to citrate, is transported out into the cytosol, where citrate is then reconverted back to acetyl coenzyme A, and this participates in the process of fatty acid synthesis. So not only carbohydrates but amino acids also contribute to fatty acid synthesis. Of course, the major provider of acetyl coenzyme A in the process of fatty acid synthesis are carbohydrates, and the other important metabolite required in the process of fatty acid synthesis is NADPH, which is also derived from carbohydrate metabolism. I believe that's an overview for fatty acid synthesis. Again, I will reiterate, do not focus on the chemistry. I just wanted to show you how the process of elongation is happening and the function of the fatty acid synthase enzyme. Please focus on the prosthetic groups, and most importantly, you should remember the COX part of the lecture, because COX inhibitors are repeatedly and regularly asked in different licensing assessments or exams. With that, let us try to end this lecture. Before that, I would like to mention that in the next session we will talk about nitrogen metabolism and the urea cycle, and the recording for that will be uploaded onto the YouTube tomorrow. Thank you very much, and thank you for listening. Tomorrow we will talk about this lecture again in the class, but we will just look at the summary form of the lecture. I will urge you to go through this lecture slowly at your own pace at home, make notes, and if you have any doubts or clarifications that you require, please approach me personally or send an email. Thank you very much.